Okay, there we go. Um, First off, I want to apologize for anyone for the confusion of the timing from this week and last week. I'm happy that we have good attendance today, and hopefully more people who couldn't make it this week are able to watch it later. My name is Mike Bell. I am an ecologist with the National Park Service Air Resources Division and a member of the NADP Critical Loads of Atmospheric Deposition uh, group, trying to figure out how air pollution is impacting the like natural resources across federal lands. This is the sixth session of our atmospheric deposition seminar series. Today we're going to have Erik Lilleskov from the U.S. Forest Service and Michaela Phillips from the U.S. Geological Survey talking about critical loads of nitrogen and sulfur for soils, mycorrhizae, and soil microbes. Like I said, this is the sixth of 7 talks. All of the previous ones are available on the NADP website, which is like with a link included in the previous emails. And next month, we are hoping to have a presentation kind of doing a synthesis of all of the talks that we've heard and how they're being used like together to inform policy and management in federal systems, as well as what the current trends in deposition are and what NADP is doing to continue monitoring these pollutants that are having an impact on ecosystems. For those who are unfamiliar, NADP is a organization based out of the Wisconsin State Lab of Hygiene. They've been monitoring deposition of air pollutants for over 43 years within 5 national networks, the largest of which, the NTN network, has about 263 sites across the US where they're monitoring wet nitrogen and sulfur deposition as well as others. Within NADP, there are 4 different science committees that use the NADP data to inform and better inform trends and patterns across the US. These are CLAD, which is the critical loads of atmospheric deposition, which this is all a part of. TDEP is the total deposition, which uses the NADP measurements alongside modeled outputs to create a more accurate deposition map across the US. AMSC looks at aeroallergens and how those are present and changing across the US, and MELD is looking at mercury deposition and mercury effects on the environment. CLAD specifically is built to facilitate technical information sharing on critical load topics with a broad multi-agency entity audience. We're not only trying to fill gaps in data, but also provide structure to how to consistently use this data to inform management and policy. We have various working groups that are pursuing all the synthesis, the uncertainty aspects, the relationship with the deposition models. So if anyone here is interested in participating, not only from a scientist perspective, but also from a user's perspective of how we can create better products to inform your decision making, please reach out and I can get you the information to get on those lists. Oh. And if you want more information on NADP, you can search the website, and there's also email listservs where updates are sent out kind of regularly. And just wanted to note that there will soon be a call for abstracts for the NADP fall meeting, which is hopefully going to be held in person in Knoxville, Tennessee, October 25th to 29th. Kind of reading trying to figure out what's going on with COVID, as well as with travel restrictions for our different agencies before that decision's made. But keep your ears out. There should be by the final seminar, we should have a decision made, whether it will be in person or virtual. And so with that, Erik, you can start loading up your presentation and I will do the introductions. So Erik Lilleskov is a research ecologist with the U.S. Forest Service, Northern Research Station's Climate, Fire, and Carbon Cycle Sciences Research Work Unit. He's based out of Houghton, Michigan, and has worked with fungi for almost 30 years, studying the response to environmental change, in particular nitrogen deposition, but also changes in CO2 and ozone. Much of his fungal work has been mycorrhizal fungi, which are symbiotic with trees and so critical to nutrient dynamics in the forest. He contributed to the empirical critical loads work for nitrogen for the US, incorporating mycorrhizal fungi into the analysis. More recently, he's also been extensively looking into the microbial role in peatland carbon cycling in a global change context. And then following him, just to get these both out of the way, will be Michaela Phillips, who is a postdoctoral scholar and ecologist with the U.S. Geological Survey's Southwest Biological Science Center in Moab, Utah. Her work focuses on understanding how global change drivers mediate plant and soil microbial community assembly in dryland ecosystems of the Southwest. During her PhD, she investigated how soil fungi are impacted by exotic plant invasion, nitrogen deposition, and fire. Her current research aims at addressing land management needs on the Colorado Plateau under the mentorship of Dr. Sasha Reed. And her work spans from examining the responses of biological soil crusts to global change to investigating post-fire recovery in pinyon juniper ecosystems. And so with that, Erik, I'll let you take it away. Oops.
Hi, can you all hear me okay?
Yep, I can see your screen.
Great, thanks for the introduction and thanks for the invitation. And because Michaela's got a really nice background in the mycorrhizal work from the Western US in the coastal systems there and the AM dominated systems, I'm going to go very lightly on them and focus mostly on ectomycorrhizal fungi with a little bit of just to touch on our vascular mycorrhizal fungi in trees, but leave the rest of it to her. And then we can fill any gaps with questions and answer at the end if there are any. So my talk today is going to try to just give a brief introduction to mycorrhizal fungi, their response to end deposition, and the implications for critical loads. Let me see if I can actually make my screen advance. Here we go. So first, I'm going to give you some background on mycorrhizas to tell you why they're such amazing organisms, symbiotic relationships. Then we'll dive a little bit into just a brief background on end deposition. I assume that 95% of you know it, but just in case there's someone who doesn't have any background, I'll give you the 1-minute version. Then go into ectomycorrhizal responses to end deposition. Look at a little bit of review of the science there, and then go to the critical load estimates for that, and including also some of the more recent European work on that's been really, I think, really groundbreaking. So first off, what are these? If I were in the room with you, you could all say, well, that's a pine seedling, right? Yeah, it's a pine seedling, and then I'd say, yeah, but, and the yeah, but is this. And the this is what's going on below ground, right? And that's the key. So this is the mycorrhizal relationship between the seedling and a mycorrhizal fungus. And really, that's what you should think about the organisms as. That's my argument. You think about a plant as it's the plant itself plus their symbionts, because it's a symbiotic organism. It's mutualistic, which means benefits both partners. And in major parts of the exchange are carbon fixed from the plants that is exchanged for nutrients in the soil that are gathered by the mycorrhizal fungi. And I always tell people that the root tips that you see there, those little blobs, I don't know if you can see my pointer, but the blobs that are at the intersection here and along the root, those are the root tips. Everything in white out here is the fungus. So the root tips are really more like a trading post for resources than they are actually uptake surfaces. And in fact, almost 100% of the uptake of nutrients surface area is provided by the fungi. And so you should think about the fungi as the primary uptake organisms, not the roots themselves, in many, and probably most plants and certainly most trees. Up to 20% of net primary production goes through mycorrhizal fungi. So I mean, they're basically getting allocated a lot of resources to do the work. So they're really important indicators of what's going on below ground. So I talked about mycorrhizae, but what you saw before was an ectomycorrhizae. So I just want to tell you briefly what an ectomycorrhizae is. I highlighted the three parts of the word so people don't get confused. Ecto is surface, means the fungus, it does not penetrate into the root cells. You can see here in these top two images here that they're going around the cortical cells and not into them. Myco is fungus and rhiza is root. The R is just there as a combiner. So there's two R's. Everyone spells the word wrong, but don't worry about it. And it's distinct from, these ectomycorrhizas are distinct from endomycorrhizas, the old term, which are mycorrhizas that do penetrate the cell wall of a root, make coils and vesicles and other features within them. Orchid mycorrhizas are vascular mycorrhizas, such as these orchid, these are vascular and orchid mycorrhizas. And Ericoid mycorrhizas are three important examples, but there are some others as well. And so these are two major classes. The organisms involved are macro structures for the ectomycorrhizas and for some of the Ericoid mycorrhizas, but not our vascular mycorrhizas. They make little spores in the soil. So any of the mushrooms, if you see a mushroom, it's associated with ectomycorrhizas, not with our vascular mycorrhizas. So I'm going to dive a little more deeply into ectomycorrhizae and the diversity. Globally, there's over 5,000 species identified, an estimated 50,000 species worldwide, possibly more. One individual host can have hundreds of potential mutualists. One study estimated over 2,000 for Douglas fir. Stand level diversity can be from the tens to over 100 species of a single stand easily. And an individual tree can form a relationship with more than 10 species. That's not uncommon to find that if you trace roots from a tree, you find that many species almost. So again, that relationship and the symbiosis I talked about is a tree plus many symbionts on its roots. It's not a one-to-one thing, so that's keep in mind. Most of the mushrooms you see in the forest are ectomycorrhizal reproductive structures, especially not decomposer mushrooms. And when I say sporocarp, it's more or less interchangeable with mushroom. All mushrooms are sporocarps. Not all sporocarps are mushrooms. Some of them are just crusts and things like that. So that's why I use the fancy lingo like sporocarp. So these are all ectomycorrhizal. But when you go below ground, that's where the action is. You saw that previous figure. That was a mycorrhiza kind of like this one here, where the root is connected to these long distance exploring types. There are some other types of mycorrhizas, the roots inside of here, and these go out and they wrap around little chunks of soil and try to decompose them. Others have very fine wefts of material that come off of them. Still others have very little material at all. And some are highly melanized, whereas most are not. And the highly melanized ones are some of the most common mycorrhizas in the world. So there's a diversity of exploration types that reflects their differences in life way and physiology and ecology. And so that sort of underpins their functional diversity. Here's a picture showing two different species on the root system of pine. Here, this is a rhizopogon species, which is related to Suillus, if people are familiar with them. And then this is a Tomentella species of crust-forming mycorrhizae. And you can see all of this white material here is all hyphae formed by this rhizopogon. And this one forms a much sparser network of hyphae. So they're different in their carbon efficiency and the amount of soil that they explore, how far they can explore from roots, et cetera, et cetera, et cetera. Also, the enzymology, these little wet drops you see here are exudates coming at in the terminal hydrophilic hyphae that are probably where a lot of the exchange goes on between the soil and the fungus. So this functional diversity is very high. It can include the pool's success, the amounts and ratios supplied to hosts. I'll give you one more detailed example in a second here. The carbon demand and allocation, as you saw in the previous picture, can be high versus low carbon demand, how much they allocate to their vegetative versus reproductive structures, And that becomes important because mushrooms are part of food webs, as I'll mention a little farther down the line. And also there are varying interspecies linkages. Some mycorrhizae may connect one species to another, even say between a birch and a pine, for example. Many of you have probably seen Suzanne Simon's work recently, the nice study she's done showing that. So I have a little power tool analogy. I know it's lame, but what it really shows you is that the tree can be thought of as sort of an organism that does one thing, which is, crank the carbon machine. You can think about that as cranking the drill, right? But the mycorrhizae can add all sorts of functionality to this system by bringing a diversity of different things they do. So the tree doesn't have to have all the functionality internally; it can rely on the symbionts. One difference first, one tree, many fungi at once, as I mentioned. So think about a power tool with many connectors to it. And the other is both partners have their own agenda. So, they're out there, they both are evolved organisms that have different things they're trying to do in this. So there's potentially for community feedback to ecosystem function. In fact, that's been shown. And just to put ectomycorrhizals in the context of all mycorrhizals from a broad ecosystem perspective, this is a figure from David Reed back in the 80s. It's a really nice sort of summary of sort of the broad patterns of where different mycorrhizal types are found. So the ectomycorrhizas are associated largely with conifer and hardwood forests, typically in boreal to temperate ecosystems in mid latitudes, where the systems tend to be a little more nitrogen limited. Sorry, there's a little pounding going outside. There's some workers outside. Hopefully it's not too disrupting. And the arbuscular mycorrhizas tend to be more in phosphorus limiting ecosystems and supplying maybe resources like nitrate and phosphorus more preferentially, although there is some diversity of that, of course. This is just a broad generalization. And then the ericoid mycorrhizas, which associate with ericaceae and heathland ecosystems, focus more intensively on mining organic matter for nitrogen. And this is a really key point that I want to make here and reinforce. And ectomycorrhizas have the ability to mine organic matter, but they vary in their ability to do that. So some ectomycorrhizas may rely more on the mineral nitrogen, others may rely on organic N. So if we go, what do I mean by relying on organic N? So the classical agricultural-based view of the soil nitrogen cycle was that the plant dies or organisms die, they drop their litter into the soil, it decomposes, goes into soil organic nitrogen forms, it's mineralized into ammonium. That ammonium might be taken up by the plant, or it could be nitrified, and then that nitrate could be taken up. The model that, as of the late 1980s, that's sort of evolving, was really a very different model. It was one that in which there's an ectomycorrhizal fungal short circuit of the soil nitrogen cycle. In this case, soil organic N can be directly taken up by mycorrhizal fungi using a suite of enzymes, including proteases. They're the tools that are used to break down proteins into amino acids, protein being the dominant nitrogen sources in the soil, but also, for example, chitinases can be used to break down chitin into their monomers, and then they can be taken up as well. And so the short circuit of the nitrogen cycle is really important in nitrogen-limited ecosystems because it means that the plants can get access to this resource competitively very effectively. There's a lot of detail behind that I don't have time to go into, but it's very important to remember that only some species are very effective at mining nitrogen from organic matter, and that, I think that has implications for the ones that are most sensitive to nitrogen deposition. So this is an example from arbuscular mycorrhizal fungi. It's a nice illustration figure. It applies to ectomycorrhizal as well, and that we show that the number of species in fungal communities, shown on the x-axis here, can affect a broad suite of traits of organisms. So from the diversity of ecosystems to biomass, shoot biomass and root biomass, soil phosphorus and pools, and also plant phosphorus, so the accumulation of plant phosphorus. So and so what you're seeing is that the mycorrhizae in these systems have a big effect on system function, and their diversity matters for that function. Mycorrhizae also are important in food webs, as I mentioned earlier. Many of you are familiar with the northern spotted owl, and it's important as a flagship species for old growth ecosystems. And there was a lot of work done back in the late 20th century looking at food webs and the importance of for example, mycorrhizal fungi shown down at the bottom here for specific food species of these top predators, such as especially flying squirrels and chipmunks and other squirrels, but also small mammals. And it's been found that if you look at the diet of these organisms, the frequency found in their diet for a number of different small mammals shown here, what you see is truffles are important for several of them, and for others, mushrooms and truffles are important as well. So you see that these become very important part of the food web for species that are also important for flagship organisms. So it's worth thinking about that as well. Think about another part of the food web, which is they are also important non-timber forest products. Humans eat them too. Commercial harvest of fungi is very important, especially in the US, especially in western states in the national forests. There's a heavy load of harvesting. Many fungi are commercially harvested. Many of the commercially harvested species are wild, are ectomycorrhizal, wild mycorrhizal fungi, such as matsutake, the Tricholoma magnivelare and Tricholoma matsutake, chanterelles, porcini, or the king boletes, and truffles, true truffles. And for example, for two of those species, the annual global trade is very substantial. So these are important economically as well. You all know the changing end cycle, so I'm going to fly through it. We've doubled rates of end, more than doubled rates of end fixation globally, and that globally doubled end fixation driven by fuel from internal combustion, point sources and non-point sources from agriculture is deposited back on the landscape as wet and dry deposition, but it's not deposited evenly across the Earth. So therefore, we need to understand the impacts in a spatially explicit manner. And here you can see that the eastern US, as you know, is highly impacted, and that the impacts are declining for most regions, except perhaps the central plains area here, where we see increases in the ammonium inputs, for example. So we've got a significant increase in the deposition from these blue and gray colors, which would be dark blue and gray, which would be sort of pre-industrial levels of end deposition, to these industrial inputs, which are significantly higher. So now that gets us to end deposition in mycorrhizae, and back in the late 90s, there was some work in Europe showing that basically fungi were disappearing from large swaths of the landscape across Europe, especially Western and Central Europe. These declines were, maybe a bit alarmist to say another mass extinction, but the decline was very, very real and very widespread. And so people were trying to figure out what was going on, and I sort of came into that mix trying to look at that too. And there were a number of hypotheses floating around for what might be causing this. So why were they disappearing? Well, first off, we saw that there were certain species disappearing and others were not. So certain very diverse genera were actually going to zero, whereas other other genera that had just a few species in them were persisting. So it was uneven among the species. That's a clue that something ecological about those species was going on. So could it be one of the hypotheses? What it could was that could be changing atmospheric chemistry that could be driving. People were looking at ozone, sulfur, acidification, nitrogen. And I focused in on nitrogen deposition as the most likely culprit and did some work on that. And most of all this work was based on mushroom production in Europe. And so there was a need to go below ground to look at what's happening on the roots as well as what's happening above ground. So when we looked at nitrogen, what we found was, and it's not just me, but a number of people have looked at nitrogen effects on fruit bodies and found that there is a decline in species diversity when you fertilize forests, and also declines in spore production for many species. But that decline, as I mentioned, was uneven. There are certain groups that were very sensitive, such as Cortinarius. And this genus Cortinarius is actually very sensitive to nitrogen and declines in production and can even disappear entirely from the forests under high nitrogen. Russula, there are many species of Russula that are highly sensitive, others that are less so. Suillus, another genus, highly sensitive. And that was what was found in the sporocarp studies. Whereas other particular groups were found to be more persistent, such as Lactarius and Paxillus and Lacaria. And so we had this sort of divergent responses. And there's this one study that I did in Alaska looking at the sporocarp response to nitrogen deposition near a point source of nitrogen. The reason we used Alaska, it's a low deposition environment, low background N deposition. We had 28 years of N deposition from a point source from a fertilizer facility. The scale was small, so we could control for other factors. And background deposition was at virtually pre-industrial levels, going up to over about 20 kilograms per hectare. So it was an ideal study system. We found 70% loss of species richness in a suite of sensitive taxa that were declining, and they matched those that were declining in Europe. And there were a few tolerant taxa that were also not declining in Europe. So this was really, again, evidence that nitrogen was a likely causal agent in the declines that we're seeing to these regional increases in nitrogen deposition. So what about below ground? When we look below ground, we look at studies that were short term, say 1, 2, 3 years in that neighborhood, very little response, very, very muted, much more muted than the fruit bodies. And that would make sense because the fruit bodies are more distal from the carbon sources, so that it's possible that they could be less responsive at the root tips. It's also possible there could be time lags in the responses. So we wanted to look long term. So again, looking back at the Alaska study where we found the sporocarp responses, we found that when we looked at the ectomycorrhizal diversity, again looking at mineral nitrogen, which corresponds to this gradient of nitrogen deposition, I could substitute through fall nitrogen going from say 1 to 20 kilograms here. We see a decrease of Cortinarius, Piloderma, some of the species that we didn't see fruiting because they make crusts below ground, and then increases in the species. Some of the species we saw fruiting, and again some of the species we didn't see fruiting again because they have cryptic sporocarps. So we see these reorganization of the communities that's consistent with what we saw above ground, as long as you have a longer enough term input of nitrogen. And the loss of species seems to be apparent at deposition levels around 5 to 10 kilograms per hectare of nitrogen per year. And then this is just showing, this differential sensitivity of the different taxa to the nitrogen availability. I don't have to go into detail, but it's the same idea. And nitrification is another strong predictor of a lot of this diversity, in addition to soil organic and. So we in the early 2000s, we took this and did a study in New England that's never, in the Northeast, has never been published. My colleague left science, and unfortunately we weren't able to complete the publication of the work, but it's still, I think, a study worth looking at because it gives context at a broader scale within the US, and there's not much information on that front. So we used a Northeast N deposition gradient in combination with a fertilization experiment on Mount Escutney to ask what's happening in a larger scale gradient with maybe more subtle N deposition gradient, and also with more landscape diversity. In that study, in a range of about 3 to about 8 kilograms of nitrogen per hectare, we did not see a really strong trend within the gradient plots, but when we added the fertilized plots, we could see a really clear trend. And it may be, and this is just an example for Cortinarius, which is one of the sensitive species in the other systems. Similar patterns show up for many of the other taxa. So we do see a trend starting to happen for some of it, and we break it down by specific nutrients. We can see some things going on, not nearly as strong as we saw in the Alaska gradient, which makes sense given the diversity of conditions, host condition and temperature, precipitation, pre-existing soil characteristics, etc. So but it does look like if you go up to a deposition of around 20 kilogram per hectare, very clearly you go down to 0. The question is, what is the shape of this function between here? Does it fall down rapidly in here, or is it more linear? The Alaska work would suggest that it goes down more quickly than this. What about in deciduous forests? So most of that work was done in conifer forests, and most of the previous studies were conifer. There were a few studies in deciduous forests suggesting less sensitivity to nitrogen deposition in deciduous forests. This study by Taylor et al. In Europe found a weak positive relationship to soil in, and Avis in temperate oak savanna it's a study with complete fertilization, including nitrogen, so it's not exactly the same as in deposition. And these levels are very high, but with that complete fertilization, they didn't find any loss of diversity. They did find a change in the community, so Quaternaria still declined, consistent with the other studies, and rustule increased. So it's possible that it's either possible these systems are less sensitive, but it's also possible that complete fertilization sort of protected some components of that diversity. Yeah, so this gets at that. There's a nice European study looking at end deposition effects on oak forests. Came out in the mid 2014, and this is using the ICP plots across Europe to get environmental data, including deposition. When they sampled among those plots, they're sort of similar to our FIA plots, but a little more intensive. And they found actually that they did detect decline in under end deposition in oak forests, looking at species richness. That decline happened somewhere around 10 kilograms per hectare of deposition up to 17. In that neighborhood is where they started seeing the transition. And there's similar species or genus level responses in terms of which taxa are the most sensitive in these systems. And actually, I think one of the really important studies that was done is taking some of that work, combining it with some other work that they've done and some new work. Martin Bader-Tondo's group in Kew looked at the response of 4 different species: beech, oak, Scots pine, and Norway spruce. And they did a threshold indicator analysis, which was a really nice method for partitioning the effects. And if you look at this middle diagram here, which is a little confusing, what it shows on the left are species that are declining, and on the right are species that are increasing under deposition at different, and shows what the centroid of their sort of abundance is. And so what you see on the left here is these little tree symbols indicate that most of the species that were declining are associated with conifers, and most of the species that were increasing are either associated with both broad host range, including conifer and deciduous, or associated with deciduous species. And so it does suggest that there's some physiological divergence among the species to do with probably the types of litter and the types of ecosystems the deciduous and the conifer species live in, that suggests that their sensitivities differ among those taxa. So the conifer systems do seem to be most sensitive, and you know, this large scale study using these indicators found a threshold of about 6 kilograms per hectare for these sensitive taxa here, in terms of indicating that would be a reasonable threshold for potential critical load as they start declining at around that level of deposition, consistent with our 5 to 10 kilograms from other studies. And so what about function? So if we look at this a little more detail, we mentioned the exploration types early on. What you can see is the species that are sensitive to elevated ends, like Swilis, Quaternarius, Phylloderma, Tricholoma. They have these very carbon intensive and dense hyphal networks, such as you see here, and either long distance exploration types or these medium distance fringe exploration types that just dump a huge amount of hyphae and sort of engulf organic matter in the soil. And they also, all of these groups are known for being very effective at mining nitrogen, with the Quaternarius being the king, actually mobilizing oxidative enzyme systems to help bust apart organic matter to get that nitrogen. So that suggests that there's both these functional reorganization and change in diversity, and there's a sensitivity of these taxa that can be pegged to nitrogen inputs. I just touch on one example from our vascular mycorrhizal fungi. Very much less work done on them, but what we see is in a gradient study in Michigan. We can see differences among the control plots with say a 3 to 7 kilograms of nitrogen in the system. So these are these dotted circles here, A, B, C, D, and that when you add nitrogen, some of these control sites start looking like the control sites for the higher nitrogen locations, suggesting that we are seeing a shift in the community as a function of nitrogen, both over the gradient and as a function of fertilization. And the exact threshold for that is unclear because it's a fertilization study, but we're somewhere in the neighborhood of less than 12 kilograms per hectare per year, or sort of expert judgment in that one. So you all know what critical loads are. These are empirically based in our case. And the metrics we use for coming up with them are loss of diversity and community change. And if those are the primary ones, and then the lesser ones where if there's no information, we also used changes in abundance or biomass of the fungi as a secondary indicator. We used long-term fertilization and gradient studies over realistic ranges of gradients and deposition values. And deviations in mycorrhizal community structure or composition from the baseline is considered to have crossed a critical load threshold. These were all incorporated into the previous critical loads work led by Linda Pardo, where Edie Allen, who is Michaela's advisor, and I were involved with that process from the mycorrhizal side. And we came up with critical load estimates for a number of different systems. I'm not going to go into all of them, but for the conifer-based ones, they're in the range of 5 to 10 kilograms per hectare, and for northern hardwoods, much fuzzier, but in the neighborhood of maybe less than 12 kilograms per hectare with a very poor threshold identified. And then for also, and Michaela probably touched on this more, but we had values from Edie's work for 7 to 9 kilograms per hectare for coastal sage scrub, and for grassland of around 12 kilograms per hectare from a Louise Erik work. So in conclusion, they're integral to mycorrhizas are integral to forest health, ecosystem health, and biodiversity. Nitrogen has a large impact on mycorrhizal fungal communities. Critical loads for nitrogen deposition are lower for mycorrhizal fungi or conifers than for deciduous trees. And more data from the US would solidify these critical load estimates. Although I would say I think the European studies and their findings probably are fairly applicable here, given the parallel genera we have there. So if it would be conceivable, we could use some of those to reinforce some of our estimates as well. Now, Mike sent us some questions. I did sort of jot down some sort of placeholder answers for those. I don't know if we want to go into those now, Mike, or how you want to handle the question and answer for this.
Yeah, you can just kind of give a quick overview of your thoughts on those, and we can dive in deeper if anyone has detailed questions for them. Okay, I'll go over this then, and then we can go from there. So the 4 questions I got from Mike were: one is, what is the value of this information called protect, preserve, or recover federal lands? Well, it provides an empirical basis for critical loads for natural ecosystems, and it also helps to understand possible causes of unexplained system change that could be mediated by mycorrhizal fungi. So, for example, in Europe, there's actually concern that nutrient imbalances in forests could be driven by changes by sort of these positive feedback loops related to changes in mycorrhizal fungal communities. That could have strong implications for forest health. So there's some work, a new paper out on tipping points, for example, that could be very relevant for thinking about. For groups working in the Pacific Northwest, for example,
if you're having problems with your Tricholoma matsutake, we know tricholomas are very sensitive to nitrogen deposition. So those matsutake, which are important non-timber forest product, could be affected by N deposition in those downwind areas from the major cities in the Pacific Northwest, for example. We could talk more about any aspects of that as you like. You might see different implications than I've sort of laid out here, but these are the broad outlines. How can a park or forest integrate their local data to better understand the risk of impacts? I think the two things you want to look at are sort of what is your local deposition? Are there hotspots on your landscape? For example, high elevation sites that might be particularly sensitive because of higher deposition, or conifer dominated, and especially when they need two of them intersect. So if you have a higher deposition and conifer dominated systems, you're likely to have more change in the system more rapidly versus deciduous forests. So that would be one starting point, I guess, is to sort of match these critical loads to for this particular factor to your system. Any data as far as is there data that a park might have that could advance a research? Any fungal community data of any sort would be wonderful if you have it, especially if you have long-term. Some people might have set up long-term mushroom production plots, for example. Anything a little more systematic, the better. But even non-systematic data could be used in some ways. I know there are a lot of groups that are doing citizen science type work where people will just go out and take photographs of mushrooms and get their locations. Those data could be mined for trying to glean fungal community compositions on a landscape because they have geolocations on those in many cases. As far as making additional research, could parks pursue to better understand the presented responses? The most obvious ones to me would be going below ground and doing a systematic network of plots that look at the fungal community on roots and tying them to any other metrics of system characteristics like nitrogen availability or N deposition or tree status. If you have FIA plots in your forest or in your park, for example, those would be good places to co-locate if you could overcome any FIA concerns. And then also putting in permanent plots to monitor fungal spore production. Sporocarps are a lot easier to access by the amateur, whereas the below ground work is pretty much for professionals. The sporocarps can be surveyed by the most incredibly avid groups of mycologists that form these local mycological clubs and societies. They go out on forays all the time. And I think if you harness that energy and said, we would love it if you would, put in plots in our forest or our park and monitor those over time, you find some very willing partners. So those could be an awesome citizen science project. Yeah, those are the things that came to mind when I did sort of a quick read through on this, but there may be others that could come up in conversation here. So with that, I guess I could switch over to if there's any time left for questions.
Oh wow, thanks so much, Erik. That was great. And some of the pictures that you had at the beginning were just phenomenal in understanding what's going on below ground. Does anyone have any questions for Erik? You can either raise your hand or just take yourself off mute and ask it directly. Or put it in the chat if you want to. As people are thinking about this, I had a couple. One is, do you have any sense of what recovery of mycorrhizal communities look like if a critical load is no longer in exceedance or as deposition decreases?
There's been some of that work, especially in Europe, it's been done, and they are seeing some release, some recovery. It varies, the rate of recovery varies, but there are lags that are likely associated with the inertia of nitrogen in the ecosystem. So, if the system is essentially eutrophied, that nitrogen doesn't go away immediately. And so it's really going to depend a lot on the rate of sort of transformations and processing of that nitrogen, whether it's being lost from the system or made unavailable in ways that might then favor some of these mining species, for example. So go ahead.
I was saying, in that recovery, do most of the mycorrhizal, are the spores still present in the system where they're able to naturally expand, or does it need to be seeded, or does soil need to be brought in from intact forest community?
I'm not aware of anyone bringing in mycorrhizae to help speed the rate of recovery. My guess is the limitation is less, depending on the scale of nitrification, right? If you've got, a whole continent, of course, then there could be limitations to spore movement. But if it's more patchy and localized, then there's obviously a spore rain coming constantly across landscapes. There are some spores that persist in spore banks for decades, much like seed banks. So Suillus and Rhizopogon species are kind of famous for that. So it's quite possible that in some of these cases, those spores are just waiting for the right conditions to germinate. But certainly, I don't know that the studies have been done to look at spore limitation versus these sort of other lags in process that are constraining recovery. There have been studies that have sort of removed the forest floor to try to accelerate recovery and they've recovered certain species, but not the ones that live in the forest floor, which are a lot of them. So it's sort of a mixed bag. Yeah.
Okay, cool, thanks. Stephanie, you can ask your question.
Thanks. Hey, Eric. So I have 3 questions. 1, does sulfur deposition affect the same way that nitrogen does? 2, do you have any thoughts on liming applications in recovery? And 3, for forest soil scientists in Region 8, Region 9, and other places in the United States that are affected by acid deposition, it's very hard for us to incorporate, mycology. And this is something that we want to do because all of us know that soil health is linked to microbes, microbial organisms, fungi, and so forth. And we've only had moderate success integrating it into any kind of monitoring that we do. Could you give us like 1 or 2 bits of advice?
Sure. So first one was sensitivity to sulfur. Did I get that right? So there have been a few studies showing some sensitivity to sulfur. It's probably not as strong. I mean, it's a pH mediated because these guys are adapted to fairly acid systems to begin with in many cases, but certainly sufficient acid deposition can send it out of the normal ranges. There's not been a lot of recent work on that, and I'm not aware of any critical load estimates for North America that have been developed for sulfur for mycorrhizae in particular. Maybe someone has seen that. I haven't, because I don't think a lot of the studies have been done. In general, the sensitivity to nitrogen seems stronger, and I think that's probably because of sort of this. It's affecting the resource that they're specifically adapted to, expecting that nitrogen, and but that resource, if that limiting resource is no longer limiting, then they're not going to be very effective symbionts. At least that's part of it. So what was the second question about sulfur?
Well, the second one was about liming. If like
liming.
Calcium, magnesium additions.
There have been a few liming studies in Europe in particular where they were concerned about this a little earlier on, and they found modest impacts of liming. It does change the communities of fungi somewhat. It won't necessarily bring back, given that the responses were a little bit muted to begin with, you may actually change the community because you're also changing calcium availability and things like that too. So you're doing a few different things other than just changing the pH. So I can definitely point you to the literature. I can point you to the European literature on that, and we can offline, if you want to send me an email, I'll give you a data dump on that. And as the third question was things you can do.
Yeah, like if you're a forest level soil scientist and you're
Right.
aware, you know, how would you incorporate this information into your field work?
Yeah, so I guess so I'm thinking about you're an NFS soil scientist and you want to know how do I manage these forests more effectively under acidification, and are you working in West Virginia, or where are you working, forest?
Well, I mean, I'm now at the Washington office, but I spent 20 years, you know, in the Monongahela, and now I'm actually with Northern Research Station. But let's just say the Central Appalachians.
Central Appalachians. So yeah, so those systems, what could you do? I think I mean, obviously you're under pressure from a lot of people to you know, make decisions that are based on the forest plan, et cetera, et cetera. So if this isn't in your forest plan, I'm not sure what you can do. If you're not don't have to monitor them or do something with them. If you have problems with forest health or recovery of a forest, for example, in a post-mining site, I was thinking about in sort of coal mining areas, there's, you know, an early literature from like Marx, et cetera, on, you know, the limitation, the benefit of mycorrhizal inoculation, for example, in those systems. So certainly the kinds of places where you know you're going to need to do something are places where you've lost the surface soil layer, and reclamation can make a huge difference. So I don't know if that answers your question or not. Yeah, and again, I'm happy to think about that with people more if there's some place on the side to do it. We do have this conversation with the Region 9 soils folks on this, so on various aspects of things they're concerned with. So if that is a concern, I'd be happy to connect with them on that topic.
Thanks.
Thanks. Well, in the interest of time, let's move on to Michaela. Thank you so much, Erik. If you have any additional questions, which I have a couple, just put them in the chat, and hopefully we can interact that way.
Yeah, I'm happy to answer anything in the chat.
Great. So Michaela, you can steal the screen.
Okay. Can y'all see my screen and hear me?
Yes.
And
yep,
it's in full screen. Awesome. Great. Well, my name is Michaela Phillips, and I'm an ecologist at the Southwest Biological Science Center in Moab, Utah, and I work with Sasha Reed. Today, I'm going to talk about nitrogen deposition effects on dryland soils. I'm going to talk a little bit about some of my PhD work and then also some of my more current work. I think this is the first talk to really focus on dryland, so I'm going to give I really like this figure. It's from Hoover et al. 2020, and it just shows just how extensive drylands are. They make up 41% of our terrestrial land surface, and they support 38% of the global population. They're also expanding, and something I think I really like about this visual is that it just shows just how diverse they are. So today I'm going to focus in on PhD work I did in chaparral systems, and then also work I've been doing on the Colorado Plateau and shrublands. And I think one of the big questions is, what do we know about nitrogen deposition effects in drylands? So more recently, there's been a lot of attention, especially in Southern California, on NDEP effects. But as you can see circled here in red, that in the Four Corners region where I currently live and work, there's been very few studies, and there's just a lot that we don't know. However, most of what we do know suggests that they're really sensitive to nitrogen deposition effects. And they're thought to maintain lower critical load thresholds. So my main questions are they actually more sensitive? And then do they respond differentially? Do those in Southern California that receive so much higher deposition rates from vehicle emissions respond differently to drylands in more rural areas like on the Colorado Plateau? And so most of my PhD work happened outside of Los Angeles. So we had really high deposition rates, and now I'm living somewhere where there's quite low rates. So first I'm going to talk about arbuscular mycorrhizal fungi, and it was great that Erik gave that really detailed introduction, because I was hoping he would, and wasn't going to go into much. So during my PhD, I worked with a colleague, Soren Weber, and we started thinking about how biomass allocation differs between arbuscular mycorrhizal families. And one of the things that I think is really important to note is that 80% of our vascular plant families make arbuscular mycorrhizal associations, and in drylands, they're very dominant. There are definitely ectomycorrhizal species, but in a lot of our shrublands, it's mostly AMF. And so the first functional grouping that we named is called rhizophilic AMF, and that is because they put a lot of allocation into interradical hyphae, or creating hyphae inside of the roots, internally colonizing the roots, and they're thought to confer pathogen protection. And then the other group I'm going to focus on today is adaphophilic AMF. So these families put a lot of effort into producing extraradical hyphae or foraging hyphae, and they're known to increase nutrient uptake as well as water. And a lot of our slow-growing shrub species in dryland systems that I focus on really heavily depend on these mycorrhizal associations, because they have very coarse roots, and they just don't have that increased surface area without them. Soren and I both wrote papers where we outlined these functional groups, and they were kind of built from existing knowledge in the literature that you can see listed there. So some of the inspiration for this work was work that my PhD advisor, Edie Allen, was involved in. And so here, this figure is from Kathleen Treseder's 2018 paper. On the y-axis, we have external hyphae length, and then we have observed soil C to N here on the right, and then on the left, the N association index. And each of the points represents a family of mycorrhizal fungi. And so these ones are adaphophilic AMF families, the Jagosparaceae and the Scutelospora, and they produce more foraging hyphae. And so in her paper, they found that these families were positively correlated with with lower soil C to N. Higher soil C to N. This backwards gets me confused. But they didn't find any relationship between external hyphae production and an association index. And then in Edie's paper here, they looked at how critical loads related between arbuscular mycorrhizal fungi and plants, and they found rapid declines in all plant and fungal values at the critical load threshold of about 10 to 11 kilograms per hectare per year. And this indicated that arbuscular mycorrhizal fungi respond to the same environmental signals as plants in Southern California, and that they could be used to assess critical loads. So both of these papers really inspired the work that I'm going to talk about that I did during my PhD. So my goal with this dissertation chapter was to determine how invasion and nitrogen deposition affects fungal community composition. And I worked at two sites. The first is this yellow one, San Dimas Experimental Forest. It was my high nitrogen deposition site. It's pretty close to Los Angeles, probably like half hour. And it's also the mountain, so it's just kind of all of the dry deposition blows over there. And they get about 19 kilograms of nitrogen per hectare per year. And the invasive grass I focused on there was the Vina fatua. It's a wild oat. And then I also focused on Bromus diandrus, ripgut brome, at a low nitrogen deposition site, Emerson Oaks Reserve, which is outside of Temecula, a little bit north of San Diego, and they receive about 6 kilograms of nitrogen, or it receives about 6 kilograms of nitrogen per hectare per year, so much lower. And at both of these sites, I looked at this native chaparral shrub, Adenostoma fasciculatum. It's one of the most common chaparral shrubs. So here, I'm just going to show one figure from this paper, just because there's a lot, and a lot of it is focused on the invasive native dichotomy, but I'll leave that out for today. So I used a hierarchical Bayesian joint distribution model to understand how these variables on the legend here affected the abundance of these different AMF families. And on the y-axis here, we have the proportion of model variance. So what this figure will show is how much of the variance in the model for the abundance of each of these families can be attributed to these variables. So this is for root data. So I looked at the composition of the vascular mycorrhizal fungi in roots. And you can see here in green that nitrate was really a large contributor, even more so than host plant, whether it was native shrub or invasive grass. And so the largest amount of the variability was attributed to the soil nitrate values, which were really different. They were much higher at our high end deposition site. And this to me suggests that the selectivity of the host plant and fungi in initiating these mutualisms really depends on soil nitrate. And that was interesting in comparison to the soil data, which really just showed kind of a contrary, that phosphorus was really important in determining the abundance of sort of all of these families. So some of the conclusions from this work, we observed relative increases in abundance of all of the families that we looked at with higher soil nitrate concentrations. However, the rhizophilic families, or those that make a lot of produce a lot of internal hyphae inside of the roots, were most positively associated with elevated soil nitrate. And these Jagosphoraceae and these other adaphophilic families showed little increase, which agreed with some previous work that I showed, the Triceder paper. And this is interesting because there is a lot of work that suggests that these foraging hyphae will decrease under elevated nitrogen deposition, which we know will be really harmful to native shrubs that have such coarse roots and don't produce these fine roots like invasive grasses. So now I'm going to shift gears a little bit and talk about more of my current work, which is focused on biological soil crusts on the Colorado Plateau. So biological soil crusts for anyone, or biocrusts for anyone who isn't familiar, is a consortium of organisms that live on the soil surface. Here in this photo, you can see mosses, lichens pictured, and they can all grow together. There are different types. They provide a lot of really important functions in our drylands worldwide. We don't have, I'm sure as many of you are familiar, we have pretty low plant cover compared to more temperate areas. And they are known to, at least mosses can fix a lot of carbon, whereas lichens can fix a lot of nitrogen. They decrease soil erosion and increase water infiltration. They do a lot of really interesting and important functions, but those are some of the most important. So when I started here in 2019, I was really lucky to come into a long-term nitrogen deposition experiment at Arches National Park that Sasha Reed maintains and set up. This experiment started in 2011, and it's been going ever since. We have 3 sites that are along a texture gradient in the Salt Valley in the park. They're all less than 5 kilometers apart, and they receive, in a full factorial design, they receive treatments at 4 levels. So there's 0, 2, 5, and 8, and this is all in kilograms of nitrogen per hectare per year. They receive this in an aqueous solution with backpack sprayers, and we apply it in the fall and the spring. So it's split up into two effective doses. This experiment is really interesting because there aren't too many long-term, or really any long-term nitrogen deposition experiments on the Colorado Plateau, and it's been just really interesting to work in the park. So I've been synthesizing a lot of the data from this experiment. The paper that is listed here also talks about the plant data, which I won't talk about too much today because I'm trying to focus on soils. So here on the y-axis, we have biocrust cover, and then on the x-axis is year starting in 2014. So this experiment started in 2011. So by 2014, these plots were had been experiencing these treatments for 3 years. And these photos show just a little bit about the composition. So these are mostly lightly pigmented cyanobacteria. There aren't very many mosses or lichens. So while super interesting because lightly pigmented cyanobacteria are very common throughout the West. It doesn't necessarily represent everything that's happening on the Colorado Plateau because we do have a lot of mosses and lichens, and I'll get into that a little bit more later. And then this photo on the right just sort of shows how lightly pigmented cyanobacteria, they have these filaments, they kind of glue together the soil. And although they don't fix nitrogen like lichens, and they can't fix as much carbon as mosses, they do provide a lot of soil stabilization and are very important, especially compared to bare soil. And so then what you see here in the figure is that we did see some differences in biocrust cover between treatments in 2014 and 2017, which interestingly were actually the years that we had the largest amount or the littlest amount of snowfall. And so I wasn't able to analyze that quantitatively, but I feel like there's more interesting things happening with snow. But these results suggest that biocrusts are vulnerable to nitrogen deposition, and something I've been thinking about is that they're perhaps experiencing a higher effective dose of nitrogen than the underlying soils, and maybe intercepting it and moderating what gets into the mineral soil. However, we didn't see any response on the plant community, and for the Colorado Plateau, for this region, the background deposition rates are around 3 kilograms of nitrogen per hectare per year, and critical loads are set to be 5, which was our medium value. So our high value is 8, which is higher than the critical load for this region, and we didn't see any effects on plant community composition or diversity long-term, but we did see these modest decreases in variable and modest decreases in biocrust cover. And then when we look at the soil inorganic nitrogen, we also see some interesting things. So first, these photos on the left are pictures of Sasha, probably really early in this experiment, setting them up. I mined on our work drive to find some old photos of her out in the field. So on the y-axis here is soil inorganic nitrogen, and in this first panel A, it's just the annual values, and we didn't see any differences until 2018, where we start to see some significant differences between the medium and the high, and this control and the low, as the star indicates. So it's really demonstrating a lack of soil inorganic N accumulation and potentially a lag effect. I'm really interested to we're continuing to maintain this project, and so to follow this and see what happens. And then this panel B here on the right, shows soil inorganic nitrogen concentrations in the short term. So this March 18th day is before we applied the treatments, and then this dotted line is right after. And so you can see there's significant differences between the red or burgundy high and the greenish low right after application, but those effects are short term and they dissipate within a month. So this kind of left us with a lot of questions. So some of the key takeaways are that that nitrogen is that added N is potentially quickly lost from the system, maybe due to gaseous losses and dissolved nitrogen losses. And then biocrust abundance appears to be more sensitive to nitrogen additions than vascular plants. However, both did not have responses at what is thought to be the critical load for this region, making me think that maybe we need to do more work here and reevaluate some of our critical loads. And it's also interesting that the soil inorganic nitrogen is not accumulating in response to it. So this left us with a lot more questions than answers in some ways, especially about volatilized losses. And so some of the things I've been thinking about since working on this paper have been inspired by Pete Homiak's work. He's pictured here. He wrote this paper in 2016 that's in PNAS, and I'll just show one figure from it. There's a lot happening here, but we'll just focus on the red boxes. So on the y-axis is nitrous oxide, and then on the x-axis is pre-wetting values, wetting values, and then days after wetting. The filled in circles are with plants and without plants. And so one of the things that he found was after wetting, and this is all in Southern California. So September would be at the end of the really, really, really dry summer, probably. Well, 2014 was drought, so definitely dry summer. And so what he found was really fast increases in nitrous oxide emissions, and also that they were attributed to abiotic processes. So this work, there's a couple other papers that he wrote surrounding this topic that got me really excited about thinking about, is that happening here on the Colorado Plateau? Are we adding nitrogen, and is it just getting blown off very quickly, or is it getting leached? And then I also, one of the things I started thinking about a lot too is in our long-term nitrogen deposition experiment, we mostly have lightly pigmented cyanobacteria. That's also what's dominant in a lot of the Mojave, especially where Pete did this work. So I was interested in if different types of biocrusts respond differentially to nitrogen additions. So that enters some work that we've just started doing. This is a pilot experiment looking at biocrust nitrogen emissions. It was really fun. We just finished it in May. It was a 5-day experiment, and Pete was collaborating on it. He came out with, you'll see photos in a minute, his really cool van setup that has a N2O and NOx box in it. His graduate student, Errol Green, who is just super impressive and great at running these instruments and doing all sorts of amazing fixes when they're not working. My advisor, Sasha Reed, and then a graduate student that I'm mentoring, Courtney Courier, who is at Arizona State, came out to be involved as well. So our objectives for this project were to identify the proportion of added nitrogen that is leaving as gaseous emissions, determine how gaseous losses vary with biocrust type, and then also uncover how nitrogen additions affect both nitrogen fixation and denitrification. So I have a keen interest in nitrogen fixation because, like I said earlier, a lot of the lichens and darkly pigmented cyanobacteria fix a lot of the nitrogen that the plants have access to here on the Colorado Plateau. So we set this experiment up in May on some Bureau of Land Management land outside of Moab, pretty close to Canyonlands National Park. And so we varied two things in the experiment. One was the nitrogen addition levels, and the other was biocrust types. We had 3 levels of nitrogen, 0, 8, and 50, and these are all in kilograms of nitrogen per hectare per year. We wanted to do 4, but we only have 9 auto chambers that I'll show you in a moment. And so we had to limit what we were able to do. And we chose 8 because that was our highest value in our long-term nitrogen deposition experiment, and where we did see some effects on biocrust. And then we also were interested in just going higher and kind of matching more of what Pete's group has done in Southern California. And then we also had 3 biocrust types. The first was bare soil. When we put these collars in, we disturbed things and made it so that it was bare. Lightly pigmented cyanobacteria as kind of our earlier successional. They're often the first to colonize an area and grow after disturbance. And lichens and darkly pigmented cyanobacteria dominated. So those we were interested in capturing because they fix nitrogen and are kind of more later successional. They take more time after disturbance to populate an area. So this is our setup. Pete has built with Errol's help this awesome van. They drove out from Riverside. It has an N2O analyzer and a NOx box in it, set up, interfaced with a Licor 8100. And then here on the right, these are the Licor auto chambers. So we had 9 of them running at once. They would close every 90 minutes. So it's pretty continuous gas flux values. We would set this up in the morning, add our treatments to each of the collars, and then let it run for 24 hours. And we also, because we were on this dispersed camping road, this photo on the left, you can see there was like a lot of interfacing and explaining what we were doing in this van covered in cardboard to the public, which was really fun. I've never had the opportunity to do science that is this public. So it was really good to get to talk to folks and talk to them about what we were doing and why biocrusts matter and why nitrogen deposition is a concern. So some of the other things that we measured N2O and nitrous oxide fluxes continuously on like 90-minute intervals. We also measured total and extractable pools of nitrogen. That's what Courtney is doing here. We brought everything out and kind of had a sweet mobile lab set up in probably the most beautiful place I've personally done lab work. We also measured denitrification potentials, nitrogen fixation using acetylene reduction, as well as microbial biomass, nitrogen and carbon. We have a lot of samples and a lot of data, and so results are forthcoming from this. I was hoping that maybe some of it would be together for this talk, but unfortunately it just happened like a couple weeks ago, and we've all been in the field, so it hasn't moved forward and yet to share results. But we're really excited to see what potential biocrusts have to interact with nitrogen deposition values on the Colorado Plateau. And so the funding from this project my project, the project I talked about here, the long-term climate manipulation project was funded by the Park Service Air Resources Division. So I wanted to thank them for that. And then a lot of the work that I did during my PhD at UCR was funded by the Center for Conservation Biology. And I think with that, I can take questions if people have them.
Awesome. Any questions for Michaela? Those continuous measurement rings look phenomenal. I'm really excited to see how you advance the work that he's done in the past. Yeah,
it was super fun to work with Pete and his grad student Errol. They are awesome, and I think I've done a little bit of flux measurements before, but the high resolution data we were able to capture, even just like getting it transferred from them to me, it's been like, whoa, this is a lot of data.
Yeah,
I'm really excited about it.
So when you were showing I think it was the second project, the BioCrust response to fertilization, there seemed to be a significant drop off in the BioCrust over the 4 or so years of the experiment. In my time in the desert, there's always been the don't bust the crust. It takes a really long time to recover. Like, is that more of that advanced lichen crust you were talking about, or will even like this drop off, can it fluctuate, get back up higher, back to that 80% quickly, or is this a long recovery process?
I think so. I've been doing some other work looking at biocrust recovery, and we're actually finding that following physical disturbance like trampling, or we had an experiment where we altered precipitation and kind of looked at what would happen if our monsoon season was a little different. We found that recovery is really rapid for lightly pigmented cyanobacteria, which is primarily what is at our site, our long-term nitrogen deposition plots and arches. And you can see that in this photo. That's sort of what they look like when they're wet. Instead of like mosses and lichens can be like really bright colored and a little bit more showy. So I think they can recover pretty quickly if they're not. One of the things we also found though was with warming, they're not recovering. So I think that can be a limiting factor, but warming doesn't seem to be totally eradicating the biocrust. It just is decreasing the abundance of some of the later successional mosses and lichens and dark cyanobacteria. Here we have seen like just kind of a slow decrease in biocrust cover at this site, which I thought was really interesting, and wasn't really part of the questions we were asking, but something I've been thinking about a lot.
Cool, thanks. There was a question from Joseph Terry asking to go to the results summary from the SoCal Chaparral study.
Yeah, thank you. It just went so fast I didn't get the chance to read it. So you
Sorry
can go
about
ahead
that. Sometimes when I'm talking, I go too fast. Working on it.
Okay, you can go to the next question. I just wanted to get a chance to copy it down. Thanks.
Yeah, and the paper from that is in fungal ecology. I saw the question, but I missed it. Hold on, let me open the chat. Have invasive grasses. So we haven't actually, one of the things that I didn't include in this talk from the nitrogen deposition experiment is we didn't see any increase in invasive grasses in our nitrogen deposition plots following the nitrogen, like between the controls and the high or medium or low deposition treatments. And that was interesting. We didn't really see any changes in plant community composition or abundance, which for me was really surprising because coming from Southern California, anywhere you go where there's a lot of nitrogen deposition, there's a lot of cheatgrass.
Are there invasive grasses present near there? There
is, yeah, there definitely is cheatgrass in that area, and there was cheatgrass in the plots. There just wasn't any difference between the controls and the nitrogen addition plots.
I have a question. Michaela, you mentioned that mosses are part of the biocrust. Are they distinct enough that you can pull them out and do elemental analysis, or are they really incorporated with the lichen and the other living things?
You definitely separate them, and there are a lot of areas on the Colorado Plateau that are moss dominated. That can outcompete the lichens and other things. Like in this photo that I showed, they're growing really intermixed, but even then you could still grab pieces of, like I've collected just pieces of just moss for vouchers or other things.
So another question with the re-emission of nitrogen, like what is the theory of where it goes? Because obviously, like eventually it has to be taken up by the ecosystem somewhere. Like whether if it's leaching out, are we seeing eutrophication in the streams, or if it's being volatilized? Does it just like spread out thinner to like a lower level where it wouldn't have an effect? Or what's the expectation of the next step once it's not taken up in this one spot where it initially falls based on the models?
Yeah, I think what we've been thinking about a lot with the sort of lack of effects we've seen in our long-term experiment, which I think something important to note is that we added really modest values of amounts of nitrogen compared to stuff that's happening in Southern California, but we receive a lot less deposition, so very realistic. But one of the things that we've been thinking about a lot is, okay, so if it's not, like we're not seeing it in the foliar chemistry, we're not seeing it in the soil, we're not seeing it changing plant communities, and the only thing that is really changing is this biocrust cover, where is it going? And so with that, I think the park is pretty interested in if it is leaching or if it is volatilizing, just because of both air quality and water quality concerns. And so I think, especially, I'm clearly with this recent pulse project we did, I've kind of think that maybe volatilization is what's happening, just because of Pete Homiak's work showing just it's remarkable how much nitrous emission, like nitrous oxide and N2O emissions there are in these dryland soils, is so much more than I would have ever expected. And I think especially in a place like the Colorado Plateau that is rural and isn't that busy, but is starting to see such a boom in recreation and visitorship, especially in the parks, where there's these long lines and vehicles are just sitting, idling for a really, really long time. I think it's interesting to think about an important, are we going to see hotspots of nitrogen deposition in areas in the park? And could could different biocrust types potentially temper those gaseous or leaching losses, or are they going to amplify them? And are we going to have potentially more air quality challenges or water quality challenges in our region? And I think that's something that some of this work was motivated by the fact that Mesa Verde National Park, I think it must have been like 10 years ago, there used to be that coal plant, and they were having really high nitrogen deposition rates. Sasha did a lot of work on this. It was like before my time here, so I can't speak too much to what the results and implications from that were. But thinking about, as we get busier in rural areas and development happens, are we going to face these severe air quality conditions if dryland soils can't hold on to nitrogen that's being deposited?
Well, thanks so much. While some of these results are leaning towards like insignificant or not seeing changes with fertilization, it's obviously less exciting from a publishing standpoint, but from a preservation of the resources, it's great to know that some of these ecosystems are resilient at higher levels than we had expected, or with measurements from other places. So it's, yeah, like you said, we need to figure out the next question to figure out where this nitrogen's going, what level is going to impact it, or is there a level that will impact it moving forward? So hopefully we can think of the next step in the next project to make that happen. Any last questions? Well, Erik and Michaela, thank you so much. This was phenomenal. Like I know the least about soils and mycorrhizae, and just great to get this great overview and level of detail. So really appreciate you both taking the time. And if anyone has any additional questions, feel free to email me and I can pass them along. And hope to see you next month. I think it's July 28th, to wrap up the seminar series.
Thanks a lot, Mike.